Welcome to the deep dive. You know those microscopic things that scientists use to manipulate DNA?
Yeah.
Get ready to impress your bio-geek friends.
Yeah.
Because today we're diving into the fascinating world of plasmids.
Awesome.
We're going to unpack this technical paper, and by the end, you'll be able to hold your own in any lab meeting, or at least win trivia night.
Uh-huh. I love it. Like you said, we're diving deep into the world of plasmids.
Exactly. So let's start with the basics. What exactly is a plasmid?
So imagine a plasmid, like a tiny self-replicating USB drive for
Huh.
genes. They're these little loops of DNA that exist inside bacterial cells, and they can carry genetic information from one place to another.
So they're like nature's couriers delivering genetic blueprints.
Precisely. And just like a courier can carry different packages, plasmids can be engineered to carry specific genes. This is what makes them so incredibly useful in molecular cloning. It's basically like cutting and pasting genetic code. We can insert a desired gene into a plasmid and then introduce that plasmid into another organism like a plant or even a human cell.
Wait, so we're talking about potentially editing the very building blocks of life here.
Exactly.
That's mind-blowing.
It is, isn't it? And it's all thanks to these tiny plasmids. This paper we're diving into today highlights 5 of the most popular plasmids used in labs. They each have their own specialties, kind of like a team of genetic superheroes.
Okay, so let's meet these superheroes. What's the first one on our list?
Let's start with PMD2G.
Okay.
This one's the expert in delivering genes into mammalian cells.
So if I understand correctly, this plasmid is the key to unlocking therapies like gene therapy.
Oh.
That's incredible. Is that right?
You got it. It's like PMD2G is paving the way for groundbreaking medical treatments.
Wow. Okay, what's next?
Give me another plasmid.
Give me another plasmid superhero.
Next up, we have the workhorse of the lab,
Okay.
PEC57. This plasmid is all about precision. It helps scientists make copies of DNA fragments and ensures those copies are accurate. Think of it like a genetic copy machine that also has a built-in spell checker.
Essential for any lab that values accuracy. It sounds like PUC57 keeps those genetic typos in check. What else we got?
Now get ready for the protein production powerhouses,
Ah.
PET15B and PET22B.
Okay.
These plasmids are the go-to choices for generating proteins in the lab.
Okay, but what's the difference between the two? They sound pretty similar.
That's where the subtle but important differences come in.
Okay.
Both PET15B and PET22B are designed for protein expression, but PET15B includes a special feature called his tag.
Okay.
This tag acts like a molecular handle, making it much easier to purify the protein you're producing.
So it's all about having the right tool for the job, even at the microscopic level. It's amazing how these small differences can have a big impact. What about our last plasmid superhero?
PET28A. This one is like a protein-pumping powerhouse, churning out copies faster than you can say gene expression. It uses a powerful promoter called T7, which really ramps up protein production.
So we've got our gene delivery specialist, our quality control manager, and our protein production powerhouse.
It sounds like scientists have a plasmid for almost every need.
Exactly. And to make working with these plasmids even easier, there are software platforms like TeselaGen.
TeselaGen, does that ring a bell for you?
It's becoming pretty popular. You see, working with plasmids can get pretty complex. TeselaGen is like having a super smart lab assistant that helps scientists design and manage their plasmid work.
So it's like having a digital co-pilot for genetic engineering. That's wild. Tell me more.
Exactly. It can automate a lot of the tedious, repetitive tasks involved in molecular cloning. Imagine you're trying to piece together a complex genetic puzzle. TeselaGen can help you find the right pieces, figure out the best way to put them together, and even check your work for errors.
Wow, it's like having a GPS for navigating the complexities of DNA. That's got to be a game changer for researchers. Absolutely. By taking care of the more routine tasks, TeselaGen frees up scientists to focus on the really exciting stuff, like interpreting the results and designing the next groundbreaking experiment. It's all about accelerating the pace of discovery.
And you know what I find fascinating about all of this? We're talking about tools that are so tiny we can't even see them with the naked eye. But they have the potential to revolutionize entire fields like medicine, agriculture, you name it.
It's true. These microscopic loops of DNA are already having a huge impact on our lives. Think about it. Plasmids are essential for producing many life-saving drugs, like insulin for diabetics.
Right.
They're also being used to develop new vaccines and therapies for diseases like cancer.
And don't even get me started on agriculture. I read that scientists are using plasmids to engineer crops that are more resistant to pests and diseases, which can help feed a growing population.
Exactly. It's like giving plants their own suit of armor against threats that could wipe out entire harvests. Plasmids are helping us create a more sustainable and food-secure future.
This is incredible. So to bring it all back, what's the one thing you really want our listener to remember about plasmids?
That's a tough one. I think the most important thing to remember is their incredible versatility. They're like tiny programmable machines that allow us to manipulate DNA with incredible precision. This has already revolutionized countless fields, from medicine to agriculture to manufacturing.
It's mind-blowing to think about the power packed into these microscopic structures.
Yeah.
We've gone from basic biology to the future of medicine and agriculture in just a few minutes. It really makes you wonder what other amazing discoveries are waiting to be unlocked with plasmids.
And this is really just the beginning. The potential applications of plasmids are constantly expanding as we learn more about them. It's a very exciting time to be working in this field.
You know what I find particularly fascinating about plasmids? It's this idea that we're taking these tools that already exist in nature and using them to solve some of humanity's biggest challenges.
Right.
Like we're working with nature now.
It speaks to the incredible ingenuity of scientists and researchers. We're not simply observing the natural world anymore. We're learning how to work with it, how to harness its power to create solutions for a healthier and more sustainable future.
It's like we're entering a new era of scientific exploration, one where the lines between biology and technology are becoming increasingly blurred. I don't know about you, but learning about all of this makes me feel incredibly hopeful for the future.
They do. Plasmids are a powerful reminder that even the smallest things can have a monumental impact. Who knew that these tiny loops of DNA could hold the key to so many breakthroughs?
It's a good thing we decided to dive deep into the world of plasmids today.
I agree. It's been a fascinating journey.
It really has. We've learned what plasmids are, explored how they're used in molecular cloning, and even met some of the most important players in the plasmid world,
Right.
like PMD2G and PEC57. Not to mention, we dove into the incredible potential of software like TeselaGen to revolutionize the field of genetic engineering.
We covered a lot.
We've covered a lot of ground, but I feel like this is just the beginning of our exploration. There's still so much more to learn about plasmids and their incredible potential.
Absolutely. And that's the beauty of science. There's always something new to discover.
So until next time, keep those brains buzzing and stay curious.